Good evening, everyone. This is Simon Fraser University's Café Scientifique, and my name is Cynthia Henson, and thank you so much for joining us tonight. At this time, we will take a few minutes or less than a minute to give time for more people to enter the room. So thank you for your patience, and thank you for joining us tonight. Again, this is Simon Fraser University's Café Scientifique, and we have a faculty member from our molecular biology and biochemistry group. And I'd like to thank you for joining us tonight. My name is Cynthia Henson, and I'll be hosting this session tonight. Thank you for joining us. We'll just give it a few more seconds, perhaps, to allow more folks to enter the room. All right, so I guess we might as well get started. As mentioned, this is Simon Fraser University's Café Scientifique, and my name is Cynthia Henson, and I'll be hosting tonight's session. I work at the Dean of Science office over at SFU, and this is a discussion series that we hold at least 6 or 7 times in a year. And we're happy to host this first session for 2024. After tonight's event, we will be sending you a feedback web survey link. We are happy to hear any comments or suggestions you might have. Also, your emails have been included in our mailing list, so you should be receiving updates on the next faculty events after tonight. I'm happy to see lots of familiar names on the Eventbrite list. Thank you so much for your continued support. And a warm hello and welcome to those joining us for the first time. It's great to see lots of community partners, even from beyond British Columbia. So thank you so much. Let me begin by acknowledging that we are privileged to gather today on the unceded traditional territories of the Squamish, Tsleil-Waututh, Coquitlam, and Musqueam peoples. We thank them for having cared for these lands and waters since time out of mind, and look forward to working with them in partnership as we continue to build this great city together. If you know whose territory you're currently on, please take a moment to reflect on this. And if you don't know whose land you're on, I encourage you to look it up and make yourself familiar with the Indigenous peoples whose land you occupy. Just a few house rules before I turn it over to our presenter tonight. You all have access to a Q&A box. The chat page has been turned off just to keep things simple. Please use the Q&A box to type in any of your questions or comments that you want us to read out to the presenter. We will try our very best to read everyone's questions and comments during and after the presentation. The live transcript is also turned on for your convenience if you find that helpful. And just a reminder to all, this event is being recorded and we will share the recording on our faculty YouTube channel. We will send that out also on an email. And it's also sitting on our website if you want to view this event and all our past Café Scientifique events. So as you see on the slide, we're privileged and happy that we have a faculty presenter from our Department of Molecular Biology and Biochemistry, Amy Lee, and her presentation title is Why Do Babies Get Sick? It's a very interesting topic for tonight. Each second 5 newborn babies die from suspected life-threatening infections somewhere in the world. Newborns and young babies are more vulnerable to severe infections than all other age groups. However, there is no fast and easy way to tell which microbes are involved, resulting in babies being both over and undertreated with antibiotics. So for tonight's talk, Dr. Amy Lee will describe how we can use genomics and machine learning approaches to tackle this very challenge. I'd like to ask you to help me welcome, Dr. Amy Lee. At this point, I'd like to invite Amy to screen share her presentation so we can begin with your talk. Thank you, Amy, for joining us tonight.
Okay, thank you so much, Cynthia, for a wonderful introduction, and thank you for that beautiful land acknowledgement.
Thank you.
We're really privileged to be here at the SFU campus. And, after Cynthia gave the land acknowledgement, I just want us to kind of really appreciate how beautiful our campus is and how wonderful it is that we have this ability to work on this campus, ask interesting questions, and tackle some global health problem, particularly within my group. So I will start by perhaps asking the audience a question. How many of us know someone who has been impacted by sepsis? And I don't know if you can raise your hands. Cynthia will keep tally for me.
Or they can just type on the Q&A box and we'll see
Maybe
it from there.
I can type in the Q&A. So sepsis is actually a huge global problem. In the last survey that was done, about 20% of global mortality is due to sepsis. And, when we look at that hard number, what that means is when we specifically look at kids under 5 years, we're talking about over 20 million cases of sepsis. And out of that 20 million, there's about 3 million deaths. Within that cohort, if you look closer, neonates have about 5 million cases of sepsis with 800,000 deaths. So neonates are defined as babies that are 28 days old or younger. So that's a tremendous number of neonates who die. And in fact, if you look at this global map that I drew, it may kind of look familiar, but looks kind of weird, doesn't it? So what it's actually showing you is the burden of neonatal sepsis globally. And you can see that the majority of the burden of neonatal sepsis are in sub-Saharan Africa and South Asia. So what is neonatal sepsis? It is a life-threatening host immune response to a severe infection. Basically, what happens is your body, your innate immune cells, recognize pathogens, and in this case, I'm showing you it can either be a gram-negative or gram-positive bacteria, and that recognition of the pathogen lead to activation of a series of immune responses that are meant to control that infection. However, that inflammatory response can sometimes go awry and go into an overdrive. And it can lead to what's known as cytokine storm, which then actually causes tissue injury and various organ damage that then lead to death. And as I mentioned in the intro, that this risk of dying is highest, especially in that first month of life. This is just another way of looking at that death. The current statistics show that neonatal deaths really account for 47% of the deaths seen in children that are less than 5 years old. And so part of the United Nations Sustainable Development Goal is to reduce that neonatal death to less than 12 per 1,000 births by 2030. So you can see where we are in Canada. We're doing pretty well. There's really less than 10 deaths per 1,000 births. In contrast to some of the cohorts I will talk about, that those are in the range of 20, with some of them potentially to 70 death per 1,000 births. So in today's talk, I'm going to talk about 3 different cohorts, one from the Gambia. I'll briefly touch upon the Papua New Guinea cohort when I talk about the Gambia cohort, as well as the Malawi cohort. So one of the questions that we have in the field is, why is the risk of dying highest in that first month of life? So what we know is that birth is basically transitioning from this sterile, protected in utero environment to a microbe-laden outside world. And there's likely a lot of immune responses that go on that need to help the infant adapt to this dramatic change from this in utero protected environment to a, sorry, the slides keep going back, to a microbe-laden world. So you can imagine that first week potentially being a very important time period in an infant's life as it's adapting to life in the outside. And for those of us who have children, we probably know the What to Expect series quite well. And, one question that we have is, well, what to expect in that first week? How do we know what's going on in a neonate's body, in a newborn baby's body? And what are the changes that go on during that first week of life that helps them adapt to the outside world? And perhaps by understanding that change at a molecular level, we can understand why infants are at higher risk of developing sepsis. So a group of scientists, and I was privileged to be a part of this study, asked this question: can we measure those molecular changes that's going on in a newborn over that first week? So what molecular changes am I talking about? Well, inside of our cells, we have DNA, and this is sort of known as the central dogma. Your DNA gets transcribed to mRNA, and this is called gene expression. The genes that are responsible for responding to the different environment potentially can be activated or downregulated. And you can sort of measure that transcription. And we'll come back to this point a bit later. And these transcripts, also known as mRNA, then gets translated into proteins. And these proteins and enzymes go on to perform other cellular functions. So as a part of this group, we decided that we're going to measure as many molecular changes as we can. And that include proteins, transcripts, We measure the associated microbiome. We measure changes, modifications on the DNA. And we measure metabolites, which is not shown here. But we also measure different proportion of cells and so on. And so collectively, as a part of this group known as EPIC, we started a huge project trying to assess what are the molecular changes that happen in an infant during that first week of life. This is all of us pre-pandemic in 2019 spring when we met at the National Institutes of Health. And EPIC is a part of the larger funded consortium at the NIH. There's a huge consortium that looks at different immune profile of humans at different ages. And EPIC specifically focus on neonates and looking at their immune responses. So the project lead is Dr. Ofer Levy, and we'll meet him a bit later. So,
Hi,
EPIC,
Amy. May I interrupt? If you could move back to the previous slide. I think it's the one before this, because there's a question on the Q&A box asking about the previous slide. There were less microbes in the C-section. I think she was referring to the image on the right side compared to the image on the left. Is that intentional and accurate?
Yeah, that very good eye and very good observation. So maybe quantity is not necessarily intentional, but you can see that there are different composition between neonates, newborns who are delivered through vaginal delivery versus C-section, in part because of their route of delivery. And so perhaps not surprisingly, newborn baby delivered through C-section acquire the microbes that are associated with them that are more like skin, right? Because this is kind of what they come in contact with, versus vaginal delivery tend to be microbes that are associated in the vaginal canal. Okay.
Thank you.
Yeah, so getting back to this EPIC consortium, our goal is to really understand everything we can about a newborn response. What does a healthy newborn look like during that first week? And can we reduce infant mortality globally through various vaccination programs? And as you can see, it's a very large consortium. EPIC stands for Expanded Program on Immunization Consortium. We are all over the world with some of us here in Vancouver. We have clinical core in the Gambia, as well as Papua New Guinea. We have teams in Belgium, and our lead, our overall project lead and principal investigator is Dr. Ofer Levy, who's at Boston Children. And you can see these are all the things that we are trying to assess. We're trying to assess molecularly what are the transcripts are changing, what are the proteins that are changing, can we measure their immune function? And I'm not going to have time to talk about the vaccine responses, but I would definitely talk about some of these additional molecular changes that we assess. So our first cohort that I'm going to introduce you to comes from the Gambia. And the Gambia is the smallest West African country, and it has a long track record in participating in clinical research. They've made significant contribution to actually development of a number of vaccines. And it also has one of the densest expanded immunization program in Africa, with a very broad range of antigens that's covered. And so we worked with the MRC unit, the Gambia, that's also a part of the London School of Hygiene and Tropical Med. What's interesting is that linguistically, at West Africa, it's highly diverse. So consent needs to happen in various non-written language. And the clinical lead of that project is Dr. Beate Katman and Dr. Buki Udoko. And they've really have recruited an incredible team working together. So part of it is engaging the community, interacting with the chief, meeting with the village counselor, and notifying the local health facilities of study. And you can imagine this is a very challenging task, right? To go and ask moms to donate their newborn baby, their precious newborn baby's blood for scientific research. Here is our project lead, Dr. Ofer Levy, at opening day, where there are talks, posters, skit, and songs, really welcoming the community and also just giving them good insight of what the study is about. There are resensitization on 2 occasions. And the first sort of cohort I'm talking about is a smaller cohort, it's 30 babies. But towards the end of my talk, I'm going to talk about this larger cohort where we're trying to recruit 720 babies, which is really no small feat. So as I mentioned, for this baby, because for this cohort, because we're really interested in understanding the changes that happen during that first week of life, 30 babies are basically recruited for this study, this pilot study. And all these 30 babies had their at-birth sampling taken. Then we randomized these 30 babies into 3 separate groups, where 10 babies then have their later day of life 1, another 10 babies at day of life 3, and the last 10 babies at day of life 7, so when they're a week old. And the reason that we do that is to minimize the number of blood draw for each infant, so that they get only 2 blood draw during that first week of life. Now, what's incredible is that because we're dealing with little infants, the little babies, we can't take a lot of blood that would normally be required for a large multi-omic study where you're trying to assess changes in the mRNA, the transcript that I just talked about, changes in the protein level, and all the immune function changes. So we had to figure out technically how to work with 1 ml blood, and this is all we got. And so from that 1 ml blood, we were able to assess 5 different molecular assays to give us a very comprehensive view of what's going on. I'm not going to have time to talk about all the different omic assay, and omic assay just means that you're measuring all the molecule for a particular within the cell. But instead, because this entire talk is mostly focused on transcriptome analysis, I'll go into a little bit more detail on how we do that, how we measure transcriptome. So as I mentioned, transcript is also known as mRNA, and transcriptome analysis really is measuring all the mRNA within the cell. And that gives us an idea of what genes are active, and it can tell us functionally what's going on in the baby. So we take our 1 ml of blood and we isolate those mRNAs of interest, and we can actually sequence them. And these sequences allow us to actually count for each gene, how many transcripts do you obtain? And we do bioinformatics that gives us a sense of what genes are active and there are more of, and what genes are less active and there are less of. And what you can see that is incredible to all of us on the team was that there is a massive change that happens. So you can see in this chart, on the first day, if you compare back to the babies at birth sampling, there's not a lot of changes. We really only see about 20 genes. But when the baby is 3 days old, we start to see 1,000 genes that's different, with about 200, over 200, that we have more of than we did when the baby was born, and 800 of them that are less when the baby was born, and so on and so forth. So you can see on day 7, this change is pretty dramatic. We're talking about over 1,800 genes that are different from when the baby was born. So can you imagine looking at a sleeping baby, 1-week-old baby? Lots of things are happening underneath molecularly. So on the right here, I'm just showing you a different way of kind of visualizing those changes. You can see when you compare a 3-day-old baby to their birth sampling, you know, there's a lot of gene changes that are kind of all connected. There's important function happening. And versus a week old, that change is even more dramatic. So not surprisingly to the neonatologists on the team, they see some of these immune pathways like interferon that's important for responding to pathogens. But the other thing that's perhaps not super surprising to neonatologists is they also see a decrease in cellular response to stress. And perhaps this is not too surprising given that birth is a pretty stressful event. And so you can imagine that maybe a day 3-year-old baby is slightly less stressed out than a birth baby.
Amy, I need to interrupt you at this part. There's someone in the audience while you were speaking about grabbing a tiny sample of blood from newborn babies. The question is, was it possible for you to access microfluidic tools? I believe I've heard they can operate on picoliters of sample.
Yes, for certain assays that is possible, but to do what we want to do, it would not be accurate enough. And so that's why we're working with 1 ml of blood. But Tom, hold on to your thought because we're going to come back to this point towards the end of my talk, where we will talk about how microfluidic tools can help us. Okay, so for those of you who love checking out TikTok and Instagram, apparently babies are not little adults. When we saw this dramatic change, what we realized is this is dramatically different from what we know from adults. In adults, if I take a blood sample from you a week apart, I'm not going to be able to detect much change. Your transcriptome is fairly stable, if you're sort of in the same physiological healthy condition. But that's not the case for the babies, right? The babies, there is massive molecular change. And in all these other molecular assays that we've done, we observe similar dynamic molecular changes. And so the takeaway is that really babies are not little adults. And there is a lot more to the eye than we know. All right. So why am I spending so much time talking about what's going on in a healthy baby? Well, this is in part because we didn't know at the start of the study that there is that dramatic of a change molecularly in an infant. By knowing that dynamic baseline, this helps us design some of our future studies, but it also helps us interpret some of our ongoing studies. So this is where I'm going to shift gears and talk about the Malawi cohort that helps us sort of ask this question: can we identify a baby who is sick, and particularly a baby who has neonatal sepsis? So as I mentioned, sepsis is this systemic immune response, typically to an infection. Now, it is particularly difficult to diagnose in infants, especially early enough in the disease process. And this is really important because neonatal sepsis can progress very fast. And we're talking about hours to mortality. And currently, our gold standard, and I'm putting this gold standard in quotation mark, and we'll come back to why that is, for diagnosing is bacterial culture. And it can take up to 48 hours. So what tends to happen is if an infant shows clinical signs of sepsis, then physicians will start antiviral and antibiotics while we wait for that bacterial culturing. And the reason for that is because prompt treatment, especially for bacterial sepsis, where you're using antibiotics, is essential to improve survival. But if you overtreat, where you give a newborn baby who doesn't have sepsis antibiotics, that can have major clinical consequences because you're disrupting their microbiome. So this sort of sets up the stage for what my lab, together with our clinician partners at BC Children, Dr. Pascal Lavoie and Dr. Constantine Popescu, are working on. Our goal is to find bacterial sepsis biomarkers that can help us differentiate this sick baby and prioritize those with really severe illness so that they can have urgent hospital admission, IV fluid, supplemental oxygen, respiratory support. This is very important, especially in the low and middle income countries, as resources are highly limited. But at the same time, what is really important for us is to find those babies who have severe bacterial infection so that we can give them proper antibiotic treatment. So how do we identify bacterial sepsis biomarker? And similar to kind of how I presented for the Gambia, you need your patient or your patient volunteer. You collect typically whole blood or some sort of tissue. You perform molecular analysis. And in our case, the molecular analysis that we chose was the transcriptome analysis that I mentioned. And you can perform various data analysis, which I'll share with you a bit later, that then help you find specific genes. And this is what we call biomarker that can separate out those infants who have bacterial sepsis versus infants who may be ill, but ill with something else. And this then can provide personalized therapy for the babies. So this cohort comes from Malawi. Malawi is one of the poorest country. It has a very low life expectancy, less than 50 years. It has one of the highest infant mortality rate. And its GDP is about 1/50th of Canada and population of 15 million. Now, I can't do this study without my clinical partner, neonatologist, Dr. Pascal Lava and Constantine Popescu, both at BC Children's Hospital. And I'll share quite a bit of work that is done by a brilliant bioinformatician in my lab, Erica Acton, who's really pushed the envelope and really pushed the study forward to help us arrive at robust markers. So a little bit more about this cohort. It's recruited predominantly from Lilongwe and Malawi. We had about 452 subjects enrolled, and you can see COVID interrupted that enrollment. Most of the subjects came from the neonatal ward, and these show clinical symptoms of sepsis. And 311 of them are less than 2 weeks old, with 90 of them that are between 2 weeks to 3 months old. And we have 51 infants who are at the hospital, but do not show signs of clinical sepsis. And they sort of serve as our control. And you'll see what I mean later on in the slides. So what are the data we collected? This is really incredible work done by Constantine. There's clinical assessment to figure out if the baby is suspected to have sepsis, here shown in red. Control babies are shown in green. We do blood culture, as that's the current gold standard. There's other clinical tests that we do. We actually we bank all the bacterial pathogens associated that grew up from the blood and CSF. We also take the blood to do transcriptome, shown as RNA-seq here. As well, we are trying to apply clinical metagenomics to see if we could improve our blood culture sampling. I won't have time to talk about this today, but I'm happy to answer questions. What's sort of neat is there is not just clinical hospitalization data, but there's also videos taken of all the infants. So we can go back and double check on our assessment. And we'll, of course, all the babies who are suspected of clinical sepsis, we provide antibiotics. The control babies, we collect their blood for transcriptome as they're needed to serve as a control. I'm going to quickly speed through this as we've seen this before, but to measure gene expression changes, you take the blood sample, and again, you isolate RNA, you put it on a sequencer, and you can count gene expression changes. And in this case, what we're interested in is we want to figure out what are genes that are more activated in the sepsis babies or differentially expressed in the sepsis babies compared to the healthy babies. So we apply machine learning, which is basically trying to find pattern in the large data that we have. And we apply a variety of machine learning algorithm. And typically how it works is you take your data, in this case, it's the expression data, the transcriptome data from the infants, and you split it into a training data set, which is 80% of the data for training data set, where you try to build your model. And 20% of your data is to test out, you sort of hold them out to test out how well your model performs. Okay, I can spend hours on this slide, but to really explain machine learning and the challenge of machine learning, I think is captured in this awesome meme, which is, do you have a Chihuahua or blueberry muffin? And I think this is at the crux of our problem for applying machine learning. Our struggle is to improve our classifications so that we can accurately pick out the Chihuahuas from the blueberry muffins or sepsis babies from the healthy babies. How then do you measure your machine learning performance? And really, I think this really accurately describes our current struggle. Okay, so one way to measure your machine learning performance is to use confusion matrix. And I think it's the best named matrix because it is the most confusing matrix to understand. But again, internet comes to a rescue. So I'm going to actually walk you through how you measure machine learning performance. Sorry, my slides keep moving as I move my mouse. Okay, so remember I told you that you split your data and some of your data goes into train the model, and then you hold out some data to test how well your model performs. So what I'm actually showing you here, you can imagine, right, is if the model predicts the baby to be septic, and the label for that baby is septic, then that's called true positive. So I'm just going to walk through the confusion matrix using the meme that's shown here. So basically, true positive, you can see the machine learning predict this woman to be pregnant, and the woman is pregnant. So that's true positive. That makes sense. This should actually say TN, which is true negative, where this woman is not pregnant, and the machine learning predict the woman to not be pregnant. Okay, so your model, you get this true positive and true negative, but at the same time, your model sometimes also screw up, as you can see from that Chihuahua muffin issue, right? And so that's where you can generate false positive and false negative. False positive is when this person is not pregnant, but is being predicted to be pregnant. Or false negative is when this person is predicted not pregnant, but obviously pregnant. And so using that combination of true positive, true positive, true negative, false positive, and false negative, you can generate an accuracy estimation for how well your model performs. And that, in fact, is what we currently have. You can look at the confusion matrix, hopefully with less confusion now. And our accuracy right now for our current model is about 80%.
Sorry
And
to
what,
interrupt, Amy. Someone's asking for you to go back to that Chihuahua blueberry muffin. I'm not sure if this person had a question about this.
okay,
It's nice to take a screenshot of this and really look through all the faces and see which one is a dog. Thank you.
sorry, so is there actually a question? Okay.
There's no question. It was just a request to go back to that slide. I guess it's
This is the best slide, I think, of my
I
entire.
agree.
Okay, so this is how well our current molecular markers perform. You can see we can set, so each of the dot represents an infant. The machine, well, the algorithm, the computational algorithm doesn't know what it is. It's sort of, we basically develop the marker and then we try to figure out how well it predict the label. And so what you can see is that we can actually separate out infants that are that have bacterial sepsis, and in orange from infants who do not have bacterial sepsis. And this is the control infants who are in the hospital for a different reason. We do have a few infant that sort of behave kind of like bacterial sepsis, but they're control babies and vice versa. And so we're, of course, needing to refine our model a bit to try to understand why that is, right? So going back to the Chihuahua blueberry muffin problem. And that's sort of where we are at now. And we're recruiting an additional cohort to really further help us build and be more confident in the genes that we find that can truly separate out those that have sepsis versus not. Okay, I'm going to quickly acknowledge the Malawi team, as this has been really couldn't have happened without all the folks around the world. And I will maybe pause a little bit to see if I can answer any questions.
Yep. At this point, I'd like to invite folks, if you have any questions or comments, please use the Q&A box to type in those comments or questions. But let me read through some. Will twin babies have exactly the same molecular changes on their first week of development?
That is a really good question. We have a couple sets of twins in the larger cohort, not in that 30 that I just talked about in the first data set. We're currently analyzing the larger cohort. So I'll have to get back to you on that.
Thank you. Next question is, you mentioned the C-section for delivery. Is it safe enough to say that babies delivered through C-section are more prone to developing infections?
So yeah, I'm not familiar enough about that to comment on that.
Okay, fair enough. Next question is, what do the changes in transcriptomic profile look like beyond the first week? Does it plateau?
You're asking a brilliant question. This is what we're trying to find out. When does it plateau? Is it after 1 week? I mean, we only sampled till the first week because that's the study design. try to convince parents with newborn to keep coming back and donate blood is pretty hard. So we don't know, and we this is something that is in planning, and some of the questions that we have. There is a companion. There's a similar study that was done. I think it was out of Sweden. And they tracked the infant out to 3 months, and their sampling is a little bit different from ours, but they see sort of dynamic change going on. So the question is, does this stop at 1 month, 3 months? It doesn't. It seems like there's still dynamic changes happening, maybe up to 3 months. But what's after that, we need newer cohort to really understand.
Thank you. I'll go back to a previous question asked that I was not able to read out. Is differentially expressed statistically significantly different, or does this just mean any difference?
All the ones I talk about are statistically different.
Okay.
Yeah.
Next question is, can you find out anything from the plasma?
Can I find out anything from, sorry?
For plasma.
Yeah, so the proteomics I didn't talk about comes from the plasma. So we really are assaying as many samples as we can find, as we can collect. It is really quite comprehensive.
Thank you. Next question is on premature babies. Can you say that these massive molecular changes also happen at the same rate for premature babies?
Yeah, so Dr. Lavoie is actually a world leading expert on that. And what they see is at birth, a preterm baby who is, basically a preterm, just definition for everyone is any infant. Full term is 38 weeks old, 38 weeks gestational age, right? So preterm is by definition any baby who is born less than 38 weeks. And preterm infants can be preterm at different stages. What's interesting is they all seem to eventually catch up, and this change, this molecular change, is pretty rapid. But we're doing similar studies in preterm to really try to define that and to get a better baseline. To really understand, let's compare a 24-weeker who is very, very preterm, 28-weeker, still pretty preterm, 32, 35, and so on. And a late preterm like 35, 36-weeker, how are they different? And so this is sort of an ongoing study question for our team.
Thank you. Did you want me to go on with more questions, or did you want to move on to your next slides?
Yeah, maybe I'll just end. I don't have too much.
Okay.
This actually kind of just links back to, we can find these markers that separate out baby right when they seem sick to figure out if they have bacterial sepsis or if they're healthy or if they're sick with something else, right? And so that molecular diagnostic can really improve sort of, rather than waiting for 40 hours, maybe we can do it much sooner. But the next question is, can we develop something that's predictive, right? Before the baby even show illness, what if there's some molecular change that you can pick up, especially right at birth, that can differentiate a baby that's going to develop sepsis later on? So I introduced you to the EPIC cohort, the pilot cohort in the first part of the talk, where we talked about 30 babies. In this larger EPIC cohort where we have 720 babies, this incidentally allow us to ask whether or not we can find molecular markers to predict from that birth sampling to predict whether or not an infant is going to have sepsis later on in that first week. And the reason why this study allows us to do that is actually the inclusion-exclusion criteria. Because we're looking for vaccine response, all babies need to be full term, they need to be healthy in order to be included in the study. However, within a small subset of this cohort, we actually have 20 infants who go on to develop different forms of sepsis, with early onset sepsis defining as sepsis that develop in less than 7 days, basically in that first week. Late onset sepsis is basically after 1 week. So we actually have about 20 infants out of that 720 cohort who go on to develop sepsis later on in that first week. And we can take their at-birth sampling to do machine learning and to see if there are things that can separate them out from each other. Now, some infants develop infection, and this can be eye infection or skin infection. These are not sepsis. Sepsis is infection, but it's a systemic infection, which means all over the blood, all over your body. Whereas the orange baby, these infections are the localized infection. And this work couldn't happen, again, with clinical expertise from Dr. Catman and a very talented collaborator, Dr. Andy Ann, who's an MD, PhD student. So Erica, together with Andy, have developed, using similar machine learning approaches, a set of markers that can separate out using at-birth sampling that predict the risk of sepsis development and infection for the infants later on. So this is quite incredible, right? Because at-birth sampling, we can predict your risk, your future risk of infection, and especially for sepsis. Then this, you can imagine, if validated in a larger cohort, will allow us to develop better at birth screening. So here, the sepsis babies are shown in red, the healthy babies are shown in blue, and those babies who just have the localized infection that are not life-threatening are shown in yellow. And you can see that it really separates out these three groups of infants. Okay, so where are we going with this? Really, the takeaway is for you to remember, babies are not little adults. They have very dynamic molecular changes that happen. We really are hoping that we can bring this to sort of a lab on the chip prototype, which I'll show you in a bit, and what that looks like. We are in the process of recruiting additional validation cohorts for any of the biomarker studies that we're working on to just really ensure our study is robust and not just restricted to the small sample that we have. We also have animal models, which I don't have time to talk about, but it gives us ways to target and direct our therapy towards. So really, our goal through improving the diagnosis of neonatal sepsis is to reduce that antibiotic use in cases where it's unnecessary. But it also, globally, can we engage health authorities to improve neonatal care by improving biomarkers, by improving our ability to diagnose those sepsis, And where the future goes is these microfluidic chips that is developed by Dr. Veras at NRC, where they've actually sent it out to space station to test, that allows you to rapidly diagnose different types of diseases. And so can we translate our work to something as futuristic as this microfluidic power blade, and really bring it to low and middle income countries to improve diagnostic. And really this all comes back to working towards the Sustainable Development Goals that's set out by UN, so that we can ensure healthy lives and promote well-being for all at all ages, and really help reduce that neonatal mortality to hopefully to as low as 12 per 1,000 live births in areas that are most impacted, which is the low and middle income countries. Okay, with that, that's the end of it. I'm happy to take lots of questions. Should I stop share?
You can leave it. You can leave it up in case
Thank my
people
team.
want you to go back to certain slides. So thank you. Keep those questions coming in. I will try to read more of your questions. Going back to that question on premature babies, are premature babies prone to get lots more diseases, would you say?
Okay, so I didn't get into this in this talk. This is something that we're working on. So full term, all the study that I've talked about right now is talking about full term and trying to study neonatal sepsis within full term infants. Now, prematurity is actually the highest risk factor for neonatal sepsis. And, typically 1 in 3, we're talking about infection risk is 1 in 3 for preterm infants versus 1 in 3,000 for full-term infants, right? And so it really is a big reason why this is also kind of the direction of what we're working on to try to tease apart is that rapid change that's going on for the preterm babies that's leading to additional risk factor.
Thank you. Next question is, can you even stop a baby from ever getting sepsis?
Yeah, so there are a lot of known things actually about that we know actually prevent neonatal sepsis. Of course, good core care and so on. Good prenatal care for moms. This is why in high-income countries in general, we don't have as high of an incidence rate of neonatal sepsis. Sorry, I'm just going back to the slide here. And of course, vaccination and breastfeeding has been shown to also reduce the risk, right? So it's a combinatorial factor. So there's lots that we can do for sure to reduce neonatal sepsis that we already know.
Thank you. And I was just about to mention breastfeeding as the next question. Does breastfeeding right from birth affect the immune development according to your study?
Yeah, so we didn't analyze, differences between breastfeeding versus no breastfeeding as a variable. We actually, at least in the Gambia cohort, if I remember correctly, have a very high breastfeeding at birth. So we didn't really have a comparator for that.
Okay. Okay. Next question. I think you mentioned earlier something about microbiota. Does this affect neonatal sepsis in some ways?
Yeah, so that's something that we're trying to understand, and we're trying to understand it from the moms, basically doing the maternal fetal dyad. And it is a very active area of research, not just by my team, but by many teams around the world.
Fabulous. Next question is, what are the common pathogens in neonatal sepsis?
So there's actually pretty big differences between high income countries versus low and middle income countries. So in high income countries in general, it's group B strep. And so this is why, at least as part of the prenatal care, right around when you're 35 weeks pregnant, they take a sample, a vaginal sample, to see if you carry group B strep. And so that's another reason why good prenatal care has really reduced the incidence of neonatal sepsis in high-income countries. Now, in low and middle-income countries, it's quite different. We're seeing a lot more gram-negative pathogens, including E. coli and Klebsiella pneumoniae. I don't have time to talk about my lab's work on Klebsiella pneumoniae, but we do also study the pathogen to try to understand, is there something special about these pathogens in low and middle income countries that lead to a more severe outcome for the infants?
Thank you. Next question is, can this research be applied to molecular changes in young children as well, or do they just apply to neonates?
Yeah, for sure. I know there are active research in those areas, and Dr. Peter Broden, for instance, you know, works on children, and so there's this type of strategies is because it provides a very holistic and comprehensive view of what's going on. Collectively, people are really applying it across all age group to try to understand.
Great, that's promising. Next question is, are there any easier sources of data that can complement blood samples? For example, only collect blood once from two babies at different stages, but maybe environmental variables at both stages for both babies and symptoms, and maybe train a model with a goal of predicting the future for babies with only one blood sample draw, and more, maybe different data?
Yeah, I think right now it's the challenge when you just take a single time point, is that you're missing that dynamic change, and that dynamic change we observe is really because we have that longitudinal sampling. We're building a model, that maybe the next time I give a cafe talk, we could talk about, but it's a very complex model that we're trying to see if we can use our current data to give us, what does a 1-day-old baby look like? What does a 2-day-old baby look like? And so on. And maybe that can allow us to address that question, if that helps.
Thank you. Next question is, when you were speaking about transcriptome analysis, can you expound a bit more about what you mean by more active genes and less active genes?
Yeah, sure. It's really quantitatively, we're saying, so when you do transcriptome analysis, you're always comparing kind of two states. You're comparing control versus sepsis, or the baby's at birth sampling versus later life sampling. And basically, all you're quantifying and you're saying, okay, this gene, there are more of it. And so maybe active is probably not the best best phrase. It's really that they're quantitatively
More.
more
Okay.
present or quantitatively less.
Thank you for clarifying. Next question is, would your model be a classifier? How much does sepsis vary in severity, and would it be valuable to develop a quantitative metric as a proxy?
Yes, do you want to come work in my lab? Yeah, we are working on severity to try to understand what's happening. So yeah, this is one thing that we're working on. And then the challenge is I'm assuming like quantitative metric, you're thinking of some sort of clinical scoring that can help the physician sort of prioritize. And yeah, so this of course is not happening in silo. I'm not a clinician. You know, I work in genomics and machine learning, and so I can't do this without my clinical partners. And so these are the kind of things that we're moving towards, but we're still quite far away. You know, I think I would say we need to build a more robust model, which means we need more cohorts.
Mm-hmm. Lots of stuff in progress. Thank you for that question. Next question is, someone wants to know more about bacterial resistance. Do these bacteria that causes sepsis process such ability?
Yeah, that is a really great question. So part of the reason why I'm studying Klebsiella pneumoniae is it is actually a multi-drug resistant pathogen. And so there is a recent study that was done by a group out of, I think they're out of England. And the consortia is called Barnard's, and they're trying to understand gram-negative pathogen that cause neonatal sepsis. And they have about 12 to 13 clinical sites across South Africa, across sub-Saharan Africa and Southeast Asia.
Mm-hmm.
And what they notice is that between 60% to 80% of the pathogens that they isolate from these septic neonates are resistant to the frontline antibiotics, the first line antibiotics that is recommended by WHO to treat neonatal sepsis, which is ampicillin and gentamicin. So the antibiotics that we're offering for the neonates are not likely to be helping or curing, right? And so these type of large cohort studies really matter because it tells us some of the current problems in our treatment as well as potential diagnosis problem.
Right, that's tough. Let me bring this next question back home, and I like this next question. Are you doing any studies relevant to Canadian babies?
Yeah, sure. So this, you know, one of the reasons why we started our study in low and middle income countries is because Canada has such a great prenatal care that the incidence of neonatal sepsis is so low. To get sufficient number of septic neonates, it would take us decades. So we started our work in low and middle income countries also because that is where 99% of the global incidence of neonatal sepsis occurs. But as I mentioned, prematurity is a huge impact to the risk factor of infants' risk factor to develop sepsis. And premature, so to bring it back to Canadian population, you know, the area that we're really focusing on, and this is in collaboration with Dr. Pascal Lavoie, is to really look at trying to understand what's going on in the preterm infants.
Thank you. I'm going to ask this next question more for the high school students who might be with us tonight. Would you have any tips or an inspiring message to share for those considering to pursue this path, this field of study? Any message that you want to share to them?
Yeah, I think ask questions, be curious um, and really understand that I think in modern science, how we can really make an impact is to collaborate across diverse fields and to really work in these large teams. Because this is sort of the way of the future, and by being able to take on interact with clinicians, I think it makes me think much more deeply about the questions I'm asking, and also, they check me, right? And they tell me, well, that is a useless model. Why did you build that? There's no clinical applicability. And so having those kind of constant discussion with people who are outside of your field really helps push you forward, I think.
Thank you. I love how you say it can be both humbling and exciting at the same time. And I like how you say ask questions and collaborate. Beautiful message. Thank you for that. I'll end with this last question, and I think it's a good way to end tonight's session. What are your next steps in this research, and how will this impact neonatal sepsis?
Yeah, so I think, our future goal is to really bring it to improve the diagnostic, and we need it to happen fast. We need it to be accurate. So some of the discussion around Power Blade, the microfluidic machine developed at NRC will really help us propel that. The other thing that's really important is, we need to make sure whatever we develop is going to work in a low resource setting. And then lastly, I think as scientists, we don't exist in silo. And I think what's really important for us is to interact with policymakers at different levels and ensuring that they get the message. We need more funding for science, but we also need help to potentially enact some of these policy changes that can help improve human health.
Right. Well, thank you so much, Amy, for sharing your research with all of us tonight. Lots of things learned tonight, and I thank you for that. And I'd like to thank all those who joined us tonight in this discussion. I appreciate all the questions and comments that you had typed in the Q&A box. I hope you can join us for the next session. It's coming up in February. Thank you so much, Amy, for joining us. But you have that information, and it will be sent to you by email. You're all part of our mailing list. So you should be receiving an Eventbrite link for this. We shift from molecular biology to financial modeling. So this is also quite interesting, from data to dollars. And we have a faculty member from our Department of Statistics and Actuarial Science. So I hope to see you then. That's happening also on a Zoom webinar format on February 27th, same time, also a Tuesday. Thank you so much, Amy, for joining us tonight. And thank you all for being with us. I hope to see you on the 27th of February. In the meantime, take care and enjoy the rest of your evening. Bye for now.